is a master's student. I'm his advisor, Margaret Hoffman. He comes to us from Cal Poly San Luis Obispo, where he actually was one of my students when I taught out there. He had lots of experience working with different tomato propagation projects. He ran the poinsettia sales, managed a greenhouse, and then came out here to start his master's. So he will be graduating this summer, and he will tell you a little bit about his research.
Awesome.
Go ahead, Jacob.
Thanks, Margaret. Yeah, so my name is Jacob, as Margaret said, and it's nice to see all these people virtually supporting the plant science seminar today. I'm excited to talk to you about some of the research that I've done for the past 2 years that I've been here involving green infrastructure. Specifically, my research looks at the role of plants and saturated zones for nitrogen removal within the green infrastructure systems, nitrogen removal from stormwater runoff within green infrastructure systems. And so that's the premise of today's talk. But before we get into talking about green infrastructure, I think it's important to take a few steps back and look at stormwater in general, understanding stormwater runoff, why it matters, some stormwater regulation, and how that ties into why we utilize green infrastructure to begin with. And then at the end, we'll discuss my experiment and some preliminary results from that. So green infrastructure, here is the technical definition from the Clean Water Act, which basically says green infrastructure is a different range of technologies that utilize plant or soil, or sometimes just the capture of water in order to assist and facilitate in enhancing stormwater pollutant or stormwater volume runoff. When we think about green infrastructure, we need to make it, I think it's important to clarify that green infrastructure isn't completely removing gray infrastructure. It's not completely taking over gray infrastructure, but it synergizes with gray infrastructure in order to deal with stormwater runoff, and it improves the quality of stormwater runoff that is flushed through our gray infrastructure systems. So what is stormwater runoff? Well, stormwater runoff is the runoff generated from rain or snowmelt events that runs over land. And so when we think of stormwater runoff and it running over land, collecting pollutants as it goes, we need to understand how we define different sources of pollution. And so when we think of stormwater runoff, we often think of it as a non-point source pollutant, which means it's diffuse, it's not coming from a single targeted area. In opposition to that, we have point source pollution, which comes from a single discrete conveyance where pollutants are emerging. The typical example of point source pollution would be a factory doing lots of, through the process of whatever the factory is doing, maybe detergents with high levels of phosphorus are in them, and the factory is flushing that directly out of the end of the factory. That is a point source pollutant. However, when we get into stormwater runoff management and the legislation around it, we actually sometimes consider stormwater runoff as point source pollutant, and it's regulated as a point source pollutant, and that's why it's important to distinguish that. And when we're looking at stormwater runoff as a pollutant, we're also looking at stormwater runoff in terms of issues with volume and pollution. And I think this diagram from the Pennsylvania Best Management Practice Manual for stormwater really shows a clean picture of what we're looking at. So in a natural system with the hydrological cycle intact, we have rainfall that's infiltrating, and we have very little runoff that's leaving the system. So with all the infiltration and all the evapotranspiration from the plants, we're getting very little runoff. Now, when we look at urbanization and the creation of all these impervious surfaces in all these different municipalities and cities, that's where we reduce infiltration, we reduce evaporative losses, and then we have high levels of runoff, and this large volume of runoff is collecting pollutants along the way, and that is when we come into serious problems. So the EPA divides the US into different ecoregions depending geographically where you're at. And these different ecoregions correlate to the general parameters we look at in the natural bodies of water, of pollutants that are permissible, or just natural levels of these different nutrients that are desirable or within range and healthy for the ecosystem. So we're looking at nitrogen. And so here we have nitrogen in our ecosystem 11. We want between 0.21 and 0.58. Now, to give reference to this, here is a composite table that was pulled from a bunch of different studies from the PA Best Management Practice Manual for stormwater. And we see here highlighted in red that lawns can produce around 1.5 milligrams per liter of nitrate, which is 300% of what we really want maximum in these natural bodies of water, in these receiving streams and rivers. So I think to really illustrate this, the Chesapeake Bay is a clear example of what happens when we have too many pollutants. So here in Center County, we are part of the Chesapeake Bay watershed, along with all these other counties highlighted in orange that drain into the Chesapeake Bay through the Susquehanna River. The Chesapeake Bay has dealt with pollutant problems for many years now. This is a diagram from the model that the Chesapeake Bay uses, that they've been using since '85. We can see that there is downward trends with decreasing amounts of pollutants, but I think it's important to look at urban runoff in particular. We've had increase in urban runoff in terms of its new pollutant loads, and that has not shown any sort of improvement. With excessive nutrient pollution loads in water systems, we get eutrophication, which is the excessive algal blooms and the loss of oxygen in water systems, which can really degrade natural water systems and also really impact the economies that rely on those. So the Chesapeake Bay has a program, and part of this program is to mitigate all this nutrient pollution in their water. And so each state that's part of the Chesapeake Bay has specific goals in place to be met by 2025. Here's Pennsylvania's goals. And right now, Pennsylvania is about 9 million pounds per year away. We have 9 million pounds per year left to reduce our nutrient load in order to achieve those 2025 goals. Now, when we look at stormwater regulation, it really all starts with the Clean Water Act of 1972, which was originally created in 1948, but has changed names a few times. And the Clean Water Act was created to restore and maintain the chemical, physical, and biological integrity of the nation's water. And this was really done through regulating point source pollutants. And that's through the EPA in most states. In Pennsylvania, the Department of Environmental Protection handles that. But what's important to note is that not only are point source pollutants regulated through this, but MS4 and construction land is also regulated through this program. And this program is the NPDES, which is the National Pollution Discharge Elimination System. And so MS4 stands for Municipal Separate Storm Sewage System. And so an MS4 is any municipality that has a separate stormwater system from our traditional systems running out of our toilets and houses, our traditional sewage systems. So MS4s are regulated through this NPDES program, which means there is regulation surrounding how we deal with stormwater. This also applies to construction of an acre or more because of the erosion and the potential particulate contamination from construction sites. There was also an amendment added to the Clean Water Act in 1987, known as the Non-Point Source Management Program. This is more of a user buy-in program where states are able to create stormwater or non-point source management programs, and then they're able to receive grant funding through this amendment. It's important to note also that part of the NPDES MS4 permitting system, it requires a management program in order to get that permit. And so when we think about management programs, that's where we actually start to get into green infrastructure and its use in this regulation. So for the MS4 permit program, there are 6 specific measures that have to be met in order to receive permitting. And we're really focused on the 5th because it deals with after construction. It deals with the regular built life of a city and the management of its stormwater. I wanted to point out that if you go onto the website, the Pennsylvania DEP website, and you're looking for resources in order to figure out how, when you're creating one of these management programs, all of the resources posted on that website deal in some capacity with green infrastructure or low impact development, which also includes green infrastructure. And I think that's an important point to make, that federal and state bodies are really pushing the use of green infrastructure in our stormwater management. So some examples of green infrastructure: not all green infrastructure is green. So this is permeable pavement, which helps diffuse stormwater volume so that it doesn't overwhelm our stormwater systems. We also have tree trenches, which can be found roadside and road medians in downtown areas that allow for water infiltration and water pollution remediation. And then finally, we get to bioretention swells, which is what we're really interested in our research, also known as bioswells. And so here we have a diagram of a somewhat typical bioswell, where the stormwater comes in, it passes through the root system, and it ends up leaving through a drain pipe. And you'll notice in this diagram, we have the drain pipe elevated where the effluent water is leaving. And so this elevation creates a saturated zone, which is another technology that we're specifically interested in my master's work. Not all bioretention swells do this. Many of them either have a drainage pipe leaving, just leading directly out without creating that saturated zone. And also we see here that there's a liner in this bioswell, and that prevents infiltration at that point. Many bioswells don't use liners, and they do allow groundwater infiltration. So here are a couple pictures I took of bioswells on campus. We are a part of a seed grant at Penn State that deals with monitoring of different bioswells across Penn State campus. Unfortunately, I wasn't able to be a part of that last summer, but this summer, this field season, I'll hopefully be able to get out there and monitor vegetation and storm events, and also do some pollinator surveys. So when we looked at the bioswell design for a little bit, and now we're looking at what can these bioswells do when we're putting them in our stormwater management plans in terms of removing nutrient loads. And remember, we said that we're interested in nitrate. So here we have bioretention reducing nitrate by 23%, which may not seem super great, but I'm going to show you a few studies in a little bit that have more promising results for bioswells. So when we think about bioswell design, this green infrastructure design, we're really looking at a variety of different parameters. But what we're really focused on, and we're going to look at some literature on, is plant selection in bioswells and how that influences nutrient removal, and saturated zones in bioswells and what that can do for nutrient removal. So I thought it'd be nice when we're looking at plant selection to first look at a study that didn't use any plants. So these are 6 different substrates that the left column shows the input of the nutrient load input into the system. And then the right column shows the output of the nutrient load. And these triangles down here show the relative difference between the two. And as we can see, apart from a little bit in sand 2 and soil 2 and mulch, we don't have a lot of significant nitrate removal in soil alone. To illustrate that point more, here we have a study that looked at 3 treatments that were unplanted, 3 simulated bioswell treatments that were unplanted, and 3 treatments down here that had vegetation with 3 different substrates. And we can see the clear difference between the efficacy of the non-vegetated versus the vegetated treatments. Here's a pretty comprehensive, detailed study looking at 20 different species of plants in terms of their capacity to filter nitrogen and other contaminants out of stormwater. And so we have the soil reference on the left and how much pollutant load that left in the effluent water. We can see a variety of different efficacy between species, and I want to point out Carex right here and how efficient that is at removing nitrogen. We see this as a trend in many studies. Carex is kind of an indicator species of being really effective at removing nutrient pollutants from bioswales, which is why we wanted to look at it in terms of a Pennsylvania water regime, a really wet Pennsylvania climate. How well does our Carex work here? Here's another look at plant species differences. And again, we have a species of Carex up here in the green, always being consistent with total nitrogen removal. And I think this is an interesting study because it really displays the point that not all species are created equal when it comes to nitrogen removal. Here we have the line of the unplanted treatment, and we actually have one species that's doing worse than the unplanted treatment, Leucophyta. So we looked a little bit about the plant component of things. Now we need to look at the nitrogen component, or excuse me, the saturated zone component of things. So this diagram may look somewhat similar. If you look down here, again, we have a drainage pipe that's elevated, which is giving us the saturated zone here. And the creation of the saturated zone allows us to create an anaerobic environment. And this anaerobic environment forces bacteria in the soil to use nitrate as its electron acceptor. And through that process, the bacteria denitrify. And at the end of the denitrification process, we end up with nitrogen gas. Now, I think this is an important area of research and be interesting to look at in terms of bioswales in the future, is that not all denitrification is created equal, right? Depending on a variety of parameters, denitrification is often incomplete, leading us with NO and N2O, with N2O, nitrous oxide, being a greenhouse gas. So I think that's important to consider for future research. The idea, again, is that we remove the nitrate from the system through this anaerobic conditions, and that is how we improve the stormwater quality. So when we see the use of saturated zones in other studies, we see promising results. And so again, you can probably guess the plant species here. This is another Carex in the green doing a good job of biofiltering, and we get significant impact though from the inclusion of a saturated zone. With these other two species, we also see drastic improvements with the inclusion of a saturated zone after they'd been growing without a saturated zone, right? A retrofitted saturated zone. It's also important to note that when we're using a saturated zone and we're using these microbial populations to remove nitrogen, a carbon source is really key. And so this, this study shows it very clearly. Down here, we have the nitrogen removal of a saturated zone treatment that doesn't have carbon. And then we look at a saturated zone with carbon. Up here, the nitrogen removal is significantly increased. Here's another study looking at depth of saturated zone with no saturated zone for nitrate being here. And we do see significant improvements in the nitrate removal of the stormwater with the inclusion of a saturated zone. All right, so we've talked about the two big key players in all of this, and that leads us to my experiment that I've been doing for my master's project. So we looked at 3 different plant species in an unplanted control. We also had a saturated zone treatment, so either the presence or absence of a saturated zone. And we also included a drought treatment at the second half of the study to see what kind of influence inconsistent watering has on nitrogen removal from a system that's been drying out for a period of time and then gets inundated with water. Can a saturated zone assist in that influx of pollutant removal? So these are our 3 all-stars here. This is Pycnanthemum virginianum on the left, Carex squamosa in the middle, and Symphyotrichum novibelgii on the right. Here's a table showing a few A few different things between a few different characteristics, excuse me, between each species. I think it's important to note native region, all of these species are native to the US and North America. And we also have wildlife benefits for all of them, with flowers being for the pollinator benefits from Pycnanthemum and Symphyotrichum, and carex is a good food source for wetland birds. So when we constructed our bioswale mesocosms, again, we really use a similar structure to the ones I've been showing you in these different diagrams. We have that elevated effluent pipe that creates the saturated zone. Our saturated zone consists of sand with a 2-inch pea gravel base. And up here in our substrate, we have a 60% sand and 40% clay soil that was sourced from a local provider. Here's our experiment all set up in the greenhouse. And now we have to think about how are we going to simulate this polluted stormwater to see how well these bioswales work at removing nitrogen pollution. And so to do that, we used weather data from NOAA to average out the last 5 years of weather data. And we came up with our wet months being June through August with about 4.82 inches of precipitation a month. We have about 12 rainfall days within each of those months, which fits nicely into a watering regime of 3 events per week. When we think about stormwater pollution, we also have to think of how much stormwater is coming into the system, and that is catchment. So how much land surrounding the bioswale or green infrastructure is actually draining into the system. And so the Pennsylvania Best Management Practice Manual for Stormwater recommends a 20% catchment area relative to the impervious surface surrounding it. And so for our experiment, we used a 50% impervious surface. Since we knew the diameter of our bioswales, we were able to back calculate and figure that the total volume for each rainfall event would be 1.6 gallons per pot. Then we have to actually think of the nutrient load we're putting into the stormwater. And so again, we're referencing that lawn level of 1.46 milligrams per liter. And so this is the stormwater concentrations we came up with for nitrate, ammonium, and phosphate. And here's the timeline of how the experiment went. We allowed the plants to establish in those bioswale mesocosms for a month before we began the experiment. Then we began that 3 times weekly application of the stormwater for 2 months. And within those 2 months, we sampled the fluent water every 2-3 weeks so that we can analyze to monitor how well the biofilters are removing nitrate pollution. After the first 2 months of the experiment, at the start of the 3rd month, we began a 2-month drought treatment, and that consisted of 3 12-day drought treatments with a 1-week buffer in between. And then whenever we re-inundated after the drought, we collected that effluent water. At the end of the experiment, we collected above ground biomass for future analysis of nitrogen and phosphorus, below ground biomass for analysis of the same things, as well as soil and root segments for potential laser ablation to look at root anatomy in terms of how the saturated zone may be affecting things. So this is what we were left with at the end of the experiment, these big bulky root systems that we washed off to look something like that. All right, so to get into some preliminary data, here I'm showing you the above ground tissue data. And here, although the standard error bars may look like there's significance in some points, there is no significance at the 95% confidence level in terms of treatment effect on above ground tissue. And we see those results for below ground tissue and total biomass. When we're actually looking at what we captured from the effluent water, how well these bioswales were actually performing in terms of nitrate removal, we're looking here at our first sample collection, and we're seeing a trend. We're seeing it very clearly where the unplanted treatment over here has high levels of nitrate effluent compared to the planted treatments. And the inclusion of the saturated zone significantly increased nitrate removal for the control, for the aster, and for the carex. So we did see benefits to the saturated zone early on in the experiment. Now, when we go to the end of the first half of the experiment, we see a little different story being told. We still see that unplanted control treatment even increasing more in its nitrate leaching into the system. The saturated zone did help the control treatment, but at this point, all of the planted treatments that did not have a saturated zone caught up essentially to all of our planted treatments that did have a saturated zone. And so after a certain period of time, we saw this drop of we saw this not drop of efficacy, but we saw this speeding up of efficacy from the non-saturated zones that matched the saturated zone efficacy. So they're kind of on par with each other. And we see that also, here's another way to look at the data between the first versus that last effluent water capture event. So we see very clearly that control. We see here that in the beginning, the aster was significantly impacted by the saturated zone, but at the end of the first half of the experiment, we don't see that significant benefit. So here's some data from our first drought, that after the first drought, we inundated it with water and captured the runoff. We see again that the controls here are high in nitrate leaching, but the saturated zones are beneficial. It's interesting to see similar trends for these drought. We would think that with a saturated zone, when we have these heavy inundations after drought, the saturated zone would show some benefit. But in all of these, the saturated zones were not significantly impactful with assisting in nitrate removal. And the droughts were significantly impactful in increasing nitrate levels in the runoff. We see the similar trends in our second drought, with saturated zone not being effective in assisting in nitrate removal. But it is important to note that that our planted treatments are doing better still at nitrate removal than our unplanted treatment that does not have a saturated zone. And so with that, we can draw some conclusions. The planted treatments were significantly more effective at nitrate removal than the unplanted control in bioswales with and without saturated zones. Saturated zones significantly increased nitrate removal in the beginning of the experiment, but after 2 months, the planted treatments without saturated zones caught up, and there was no more significant impact. When we're looking at the drought had a negative impact on all the treatments, and saturated zones were only really effective, significantly effective, with our unplanted controls. And so to summarize all of this talk, we've talked about how urban stormwater runoff is a real problem that impacts natural bodies of water and eutrophies natural bodies of water. Bioswales were shown in the literature to be functionally effective, and in our research to also be functionally effective at removing pollutants from stormwater runoff. It's still, we've seen saturated zones be effective, like in the beginning of our experiment, but it's up in the air right now in terms of how effective saturated zones will be in the long run if we implement them out in the field. And we need to do more research on how saturated zones are functioning within these systems. For future outlook on research in green infrastructure and bioswales and nitrate removal, I think it's always important to do more species trials with green infrastructure, specifically to your environment and climatic conditions. Pennsylvania is a high stormwater, highly moist environment. And so it's important to see how our plants are impacted by that versus how other studies plants deal with nitrogen for their potentially drier climatic conditions. Margaret's other graduate student, Tim, did some research, and he gave a talk about 2 weeks ago on his research with road salts in bioswales. And so I think it would be interesting to collaborate, seeing how road salt would influence and move in a bioswale system that had a saturated zone. So the road salt is not able to as easily infiltrate past the root zone. And again, I think it's important to also keep in mind delving into more of the microbial impact in green infrastructure and seeing how we can change that ratio of complete to incomplete denitrification. And so with that, I'd like to thank my advisor, Margaret Hoffman, my committee, Jim Selmer, Hong Wu, and Laura McPhillips, my cohort, Tim Gilpatrick, who's pictured here on the right, helping me mix soil, and Lisa Nguyen for all their support. And special mentions to the Lynch Lab for all of their endless support with equipment, with lab space, and so much. And also the Soil Research Cluster Laboratory headed by Dr. Govere, who helped me a lot with understanding the use of instruments for analytics. So thank you, and I'm open for questions now.
Any questions for Jacob?
I actually have a quick question.
Yeah.
So thinking about climate change and an increased potential for extreme rain events, are there any characteristics of these bioswales that are like, how would you change what you're doing if we're having more extreme rain events, or do you think that would change your results at all? Is that a reasonable question?
I think so, yeah. Well, I think that was kind of what we're looking at with this experiment. If we're seeing these more extreme droughts, and then there's studies that look at the excessive drying and then re-wetting of substrates and green infrastructure and how that impacts nutrient removal. And so, so that was kind of what we were looking at in this study, seeing if we do have these more extreme droughts and more extreme rainfall conditions, is there any benefit to the inclusion of a saturated zone in an environment that's already really moist? And so the kind of idea is, I don't know if this is exactly answering your question, but a lot of research on saturated zones is in more xeric climates, like in Australia, where the saturated zone is in a really dry condition and it can actually help with plant establishment and things like that. So when we're looking at more wet conditions where we already have high moisture content in our soils, we're trying to rationalize, is there any way that the inclusion of a saturated zone will be beneficial? And potentially, when we have these more extreme rainfall conditions, and we have this excessive drying and then re-wetting of the soil, we often see more pollutants come from that, come out of soils that have been subjected to that kind of drying and re-wetting. And so the idea was hopefully that we'd see some promise with saturated zones helping in uptake of those more excessive nutrients. Did that answer your question, kind of?
Yes, yes. I think that I have a limited understanding of the physics of kind of, I was imagining, well, if you get way more rain coming in, does it just do you lose more of that water? And so I just don't understand what the bioswale does with excess water that comes all at once.
Yeah. So bioswales are really varied in how we construct them. And so if you know the hydrological, right, if you know the climatic conditions, or you can predict the climatic conditions, you can create a bioswale that's large enough with a large enough saturated zone to handle all that influent water.
Gotcha. So you might need to make bigger bioswales if we have more intense rain events in the future.
Yeah.
Okay.
Yeah, that's one way to look at it. Also, the depth of the bioswale and the depth of that saturated zone to hold more water as well. And then the water ends up leaving the system depending on if, right, when we include an internal water storage or saturated zone, excuse me, the water doesn't infiltrate. And so that leads the system through a drainage pipe. That drainage pipe could lead to more green infrastructure, or it could lead to gray infrastructure after it's been cleaned through the bioswale.
I have a question that's related to that. And I think the literature suggested this, but maybe you can clarify. So if it's saturated for a certain amount of time, will there be more nitrous oxide, released from that environment of anaerobic conditions? It looked like some of the literature indicated there were more NOx emissions from those kinds of systems. Is that right?
Yeah, so I haven't delved super deep into the microbial component of this, looking at the actual ratios of what's released from the systems, but so I can't give you a clear answer on that. Yeah, sorry about that.
I wonder if that could be a reason where you sometimes didn't see such big differences among some of your treatments, because maybe there wasn't less leaving as nitrate, but there was more leaving as N2O, or is it, I mean, did you have an interpretation for why you didn't always see those expected differences?
Yeah, so are you talking about here in our drought treatments where we didn't see impact from that saturated zone? Is that what you're referencing?
Yeah.
Yeah. So one part of this is we've just barely started digging into the data. And so we're also going to get the nitrogen and phosphorus data from all the other components of these biosoil systems. And the idea is to do an averaged mass balance analysis. And using phosphorus as an indicator, because it sticks into systems more than nitrate does, we can see how efficient we are at that mass balance analysis. And then we can be pretty confident that if we don't find the nitrogen in the plant or the soil or the effluent, that has left the system as in a nitrogen gas form.
So that's another question. Are the plants harvested at any point to remove nitrogen or nutrients from that system, like at the end of the season? Are they just from a perennial habitat, perennial plant management system, are they removed?
Yeah. So general management guidelines vary depending on where you live, of course, but generally depending on what you're growing in the systems, there is management and removal of the above ground biomass with a lot of the perennials. And that's usually once or twice a year, yeah.
And so the last question would be, are these bioswales often planted with just one species as you did in the study, or do people often plant mixtures of species?
Yeah, that's a great question, a great point. Traditionally, bioswales and biofilters used to just be grass, but now there's more interest in multiple species dynamics. And that's another way that this research can go, is looking at multiple species composition and that effectiveness. But yeah, bioswales are leaning now towards a more holistic approach, not just thinking about the nutrient removal from the systems, but also what can these plants and this biodiversity do for the surrounding environment.
Thank you.
Mm-hmm.
Good seminar, Jacob.
Thank you.
I wanted to ask you, between the species, did you see any differences? Were those significant? And would you favor one species over another, or recommend one for one particular reason over another one?
Yeah, so when we look at the species, we would expect Carex to do the best because of all that literature that I was showing you with how effective Carex is. But actually, we didn't see significance between species in terms of nitrogen removal. So in this instance, we don't favor one species over the other. But again, we have to look at it in a broader context. This was a relatively short experiment. How would these plants handle long-term establishment in these systems?
Can you comment on what would be species characteristics that would be best for this removal of nitrate?
Yeah, there have been quite a few studies that look into that. And so a lot of the characteristics that have been looked at are root characteristics. So the average root length, higher average root length corresponds to higher removal of nutrients. Also higher root mass in general corresponds to higher removal of nutrients. And so we see a lot of correlation between root mass and nutrient removal from systems, definitely.
Thank you.
Mhm.
Any other questions for Jacob? All right, well then let's thank Jacob, and then I guess we're gonna open it up and have a little coffee discussion period for everybody.
Thank you, everyone.
Nice job, Jacob. Thanks.
I'm gonna stop the recording.